So let's look at the nervous system. And the nervous system is the fast signaling system of the body that is specialized pulses from one area of the body to another. And we will see that we will receive lots of information from the periphery, that is about our senses, via our nervous system. And that information will be brought to the central processing area, that is our brain and spinal cord. And our brain and spinal cord will use this information to make decisions and then send commands, electrical impulses, out to effectors, such as things like skeletal muscles or smooth muscles, glands, cardiac muscles, adipose tissue, and the like. Now, the main type of neuron that we have or the type of cell that we have in the nervous system is the neuron. We've seen it before in lab. We've seen that it has an axon, a single axon that emerges from a cell body, and oftentimes several dendrites. We can have one to several dendrites that are the receiving end of the neuron. And these dendrites will receive signals either from other neurons or from receptors that are going to give us information about what's going on. Now, the cell body will integrate all of this information, decide what to do with it, and if it wants to send a signal to another neuron or to an effector, it will then propagate an electrical signal that we call an action potential down the axon. So the axon carries the action potential away from the cell body. And this is important because this is how neurons will propagate signals from themselves to other neurons and to effectors. We also see that we have these things called glia, sometimes glia, but we hear them termed neuroglia or neuroglia or neuroglia. And these are supporting cells that help maintain the nervous system. And we will see that these cells have a variety of functions. And we'll look at some of those functions in just a little bit. But now let's break the nervous system down into its two major divisions, and then we can look at some of the subdivisions. So here we have the two major divisions, the central nervous system, which is here in gray. That is the brain and the spinal cord. And that is the central nervous system. This is going to be the area that's going to integrate information and then decide what to do with it. Then we have the peripheral nervous system. And the peripheral nervous system is everything outside of the central nervous system. And basically it is a series of two-way information highways, if you will. So basically with north and south lanes that will both bring information from the periphery, from sensory receptors, to the central nervous system. And then once the central nervous system decides what to do with that information, we'll send commands to effectors that will then do something about that information. So for example, let's say you put your hand on a hot stove, and then that information comes to your spinal cord, which is hot stove, and pulls the hand away immediately. So we have, in this case, a very simple, what we call a reflex arc, where we have a sensation that is brought to the central nervous system via this peripheral nervous system. And then the central nervous system gets that information, decides what to do with it, and acts on it by sending commands, again, via the peripheral nervous system, to effectors that are going to move that hand away from the hot stove. So when we look at our peripheral nervous system, we know it has two major divisions. It has the sensory division, which is going to be bringing information from sensory receptors. And this is going to be what we call the afferent division. And afferent means bringing towards. So we're going to bring information about our senses from the peripheral nervous system to the central nervous system. Then once it gets to the central nervous system, in a lot of cases, it may reach our conscious awareness. It may not. But in any case, our nervous system will decide what to do with this information and if any action is necessary. And if an action is necessary, so for example, in our withdrawal reflex, pulling our hand away from the stove, the central nervous system realizes that this is a noxious stimulus that could burn our hand and cause damage. So it will send commands via the motor division, also called the efferent division, because it means to send signals away from. So the efferent division, in this case, efferent means away from, it's going to leave the central nervous system, that information, these commands, and go towards effectors, which in the case of our withdrawal reflex is skeletal muscles. So what we will see is that we have information coming into the central nervous system via the peripheral nervous system, and commands issued by the central nervous system going out to effectors will also be carried by the peripheral nervous system. So that's why we have these two divisions, the sensory division and the motor division. So we can think of the efferent motor division exiting the central nervous system and innervating an effector. An effector is something that does something. So skeletal muscle is an example of an effector. A sweat gland is an example of an effector. And when we saw in that example in the very first lecture, we saw that the hypothalamus, which is in the brain, will sense when the body temperature is rising or falling, and it will do something to counteract that temperature change via negative feedback. And so in the case of your temperature rising, let's say you're going outside on a hot day and doing some physical exercise, then what will happen is the hypothalamus will sense that because information is being brought from the periphery. Blood flow is coming through the hypothalamus, and the blood is actually getting quite warm, and there are sensors for temperature. And that will tell the hypothalamus that we've moved away from our set point, our idealized body temperature. And so the hypothalamus will issue commands that will then exit the central nervous system and go to effectors. In this case, it will be a sweat gland in that area or sweat glands all over the body. And also blood vessels near the skin. It will dilate those blood vessels to bring more blood to the surface where it can radiate heat away as the sweat is perspired away from the body. So an effector is something that does something. And a sensor is something that brings information about something. So once again, our sensors come in by our afferent division, our sensory division, and that's going to travel towards the central nervous system. And for this reason, we also call it ascending pathways because it ascends from the periphery to the central nervous system and even to the brain, which is the main integrating system or the main decision-making system of the central nervous system. And there, decisions will be made, and then commands will be issued to effectors to do something. So the effectors are things like skeletal muscle and for our conscious control. And then we have our autonomic nervous system, which operates under unconscious control. So our motor division then has two subdivisions. Our somatic nervous system, which controls skeletal muscle. This is mostly under our command. So this is mostly under our conscious command. Then we have the autonomic nervous system, which is not under our conscious command. This would be, for example, like the example of the hypothalamus issuing commands to sweat glands and endothelial cell walls of these blood vessels to dilate so that we can sweat and perspire away this excess heat. So the autonomic nervous system is not within our conscious control, and it will control things like smooth muscles, cardiac muscles, glands, even adipose tissue. So the autonomic nervous system controls things like heart rate, breathing, things that we wouldn't want to have to think about all the time, because if we did and we got distracted and we stopped breathing or our heart stopped, we would die. So obviously the autonomic nervous system takes care of the day-to-day things that we don't want to have to spend our resources or energy thinking about. Now let's look at some of the cells within the central nervous system. We'll start a little bit with the central nervous system here, and then we'll talk about the peripheral nervous system a little bit more. Before I move on, I will say that the peripheral nervous system is contained, or I should say comprised of 12 pairs of cranial nerves that come off of the brain and the brain stem here, and then 31 pairs of spinal nerves. And these will then give rise to all kinds of nerves that will supply the rest of the body. So the peripheral nervous system has 12 cranial nerves and 31 spinal nerves. All right, now let's look at some cells within the central nervous system. So we have several types of neuroglia. So we've got neuroglia being the supporting cells. Now, one of the things we talked about a little bit earlier, and I'll go forward a little bit here. Here is our neuron cell body. Here we have our chromatophilic substance here that gives it its color. And neurons, if you look at neuron cell bodies and dendrites, they look gray. And hence we call them gray matter. If we look at the axon, some axons are what we call myelinated, and that's they're wrapped around by cell membrane of certain cells that provide this myelination, and that acts like insulation. And that allows the axon to conduct the action potential faster. So if we go back here, we can see that in the central nervous system, that myelin is provided by something called an oligodendrocyte. And oligo means many, or means few. Actually, it myelinates a few of these axons. And it also, dendron has to do with tree, and cyte is a cell. So if the term oligodendrocyte sounds very difficult, just simply break it down into oligo, a few, dendron, tree, and cyte. So this cell, cyte is a cell, is basically going to give off processes like tree branches that will then wrap their cell membrane around an axon. And since in the central nervous system, one of these cells will myelinate segments of multiple axons, we call them oligodendrocytes. In the central nervous system, we see the oligodendrocyte is responsible for myelination, where when we get to the peripheral nervous system, we'll see that there is another cell type that provides myelin for axons of the peripheral nervous system. Now, we also have this guy right here, a microglia or a microglial cell. Microglia is the term we usually hear most often, but either way is fine. And these are like little phagocytes. These will go and pick up cell debris and dead stuff and stuff you don't want there anymore. The detritus from the goings on in the central nervous system. So these are our scavengers. Then we have our astrocyte. And our astrocyte has several functions, one of which is to regulate the interstitial concentrations of certain ions. And this is important because, as we know, neurons are electrically excitable cells, very similar to what we saw with muscle cell. And there are some differences, which we'll cover in just a little bit, but we need to make sure that the ionic concentrations are tightly regulated so that the number of ions, or the type and amount of ions outside the cell, is maintained where it's supposed to be, as well as the type and number of ions within the cell. And as you recall, we've got a lot more sodium outside the cell and a lot more potassium inside. We saw this with muscle cell, and we'll see it's true with neurons as well. Now, the astrocyte also has another function, and that is to wrap these processes, called astrocytic infeed, around blood vessels, especially capillaries. And as you remember, capillaries are basically these very, very small blood vessels that have only their tunica intima left, only their innermost layer. And this is comprised of the endothelial lining and not much else. And so these interstices between the endothelial lining is where stuff can get through. And in the brain, we don't want certain chemicals to get through. So we have these astrocytic infeed that wrap around the endothelial cells, and they regulate the tightness of these little interstices here, so that things that might be able to percolate out of circulation in the rest of the circulatory system will not be able to percolate out in the brain. And this becomes very important, or in the spinal cord for that matter, because we will see that things like glutamate, which is an amino acid, is also a neurotransmitter. So while glutamate is an amino acid that could be used by the body to build proteins, we don't want it in high concentrations in the brain because it is one of the chemicals that will facilitate signaling between neurons. And so if we had a bunch of free glutamate in the central nervous system, it might promote signaling between neurons where we don't want it. And it could even overactivate neurons and cause what we call excitotoxicity. It could overexcite neurons. So we want to keep certain stuff out of circulation in the brain that in other parts of the body really doesn't make that much difference. Now, we also have one other type of glia called an ependymal cell. And this is going to make something called cerebrospinal fluid. And cerebrospinal fluid will fill areas in the brain called ventricles, and also an area of the spinal cord called the central canal. And then we're going to have a series of coverings of the brain and spinal cord called meninges. And the cerebrospinal fluid also flows through that. And we'll see that the cerebrospinal fluid has many important characteristics and properties for the central nervous system. And so the cerebrospinal fluid, as the name implies, is a fluid that's in circulation within certain areas of the brain called ventricles, and also in certain areas of the spinal cord, what we call a central canal, as well as the meninges, or the coverings that cover all the tissues of the central nervous system. Now, let's have a look at a neuron. Here's a neuron. Once again, we have our cell body that we sometimes call the cytoplasm, perikaryon, and we have our chromatophilic substance here, which gives it its gray appearance. A lot of times you'll see little rosettes of rough endoplasmic reticulum and ribosomes because there's a lot of protein production going on in the cell body. And this will give this neuron a dark staining color. So once again, our cell body and our dendrites will have this gray appearance, whereas our axon, especially the myelinated axons, myelin is mostly lipid. And so lipid is not a good conductor of electricity, so it acts as an insulating sheet, and it will constrain the electrical signal to stay inside the axon rather than jump off and maybe arc somewhere else. So it's very similar to putting coatings like Bakelite or plastic or rubber coatings on electrical wires to prevent the signal from jumping. And in this case, it allows the signal to be conducted much, much faster. Now, one of the things we will note about the neuron is we have the integrating part of the neuron is the cell body. We have the dendrites, again, are the receiving end of the neuron. They are going to receive signals from other neurons or from sensory receptors. And then that information that is conveyed via the dendrites will be integrated by the cell body. And then if this neuron is going to send an electrical signal either to another neuron or to an effector of some sort, then it will send that signal as what we call an action potential down the axon, which leads away from the cell body. And the axon, once again, in a lot of cases is myelinated to increase the velocity with which that signal can be conducted. Now here we have an axon that is in the peripheral nervous system. And we know this because it's a slightly different kind of cell. Instead of having an oligodendrocyte, which will myelinate several segments of a few axons, we have one cell myelinating exactly one segment of one axon. And these are called Schwann cells. And here's a nucleus of a Schwann cell. And what you see is what's happened here is it has wrapped itself around this axon. And very much like a carpet, rolling up a carpet, is rolled up this bunch of membrane bilayer, which is all phospholipid, and has squeezed all the cytoplasm to the outside. So it's very much like if you had a rubber raft and you're deflating it and you're rolling it up and you're pushing all the air to where the air valves are. But everything else now is just a layer of polyvinyl chloride or whatever your raft is made of. So basically it's very much like that. It's like rolling this thing up around the axon and the cell. So the cell membrane is being rolled up around this axon and squeezing the cytoplasm to the outside, so that you have this little bit of cytoplasm with the cell nucleus on the outside, but all of this is just phospholipid bilayer with nothing in between. So almost all of it's lipid. And lipid is not a good conductor of electricity, so it insulates this axon. And we will see that we have these nodes between these segments that are myelinated. And these are called nodes of Ranvier. And then the segments that are myelinated are called internodes. And these are where we will see that the electrical signal is constrained to stay along the axon, and we will see that the electrical signal is regenerated at the nodes of Ranvier. And we'll see how that works a little bit later. Now let's zoom in on the Schwann cell here, and you can see how all we have is this phospholipid bilayer wrapped around this axon. And all the cytoplasm has been pushed to the outside. So here we see some of our cellular organelles here, and we see our cell nucleus. And we have a special name for this outer covering. We call this the neurolemma. And if you remember from sarcolemma, lemma means sheath. So this is sort of the sheath of the axon, especially the myelinated axon. So the neurolemma is the sheath of a myelinated axon into the outer part of the myelin sheath. Now, just like we had for muscle, we have special terms for neurons. So we have within the axon, the cytoplasm within the axon is called the axoplasm, not surprisingly. The cell body of the neuron, because it has this large cytoplasm that surrounds the nucleus, we call this the perikaryon. And karyon means nucleus, and peri means around. So we have a term for that, perikaryon. And we have other terms that we'll see that relate to the neuron. And we will see that just like muscle cells, it is an excitable membrane. So this thing can conduct an electrical potential. And in the case of the axon, we can conduct an action potential. And in the case of the dendrites and the cell body, we have a different type of electrical signal that we will see that is not an action potential, but basically what we call postsynaptic potentials that are graded. And what we mean by graded is that their amplitude, or that is their shape, how big they are, is going to depend on how much that axon, that dendrite is stimulated, or how much the cell body is stimulated. And we'll look at the mechanism of that a little bit later on. But we have what are called graded potentials and what we call postsynaptic potentials all along the dendrite, because we are receiving signals from somewhere else, from axons of other neurons or from receptors, sensory receptors. But if this cell decides to fire an electrical signal, it will do so along its axon. And the axon leads away from the cell body and propagates what we call an action potential. And the action potential is very much like an all or none binary 1 or 0. So either you're going to have an action potential or you're not going to have an action potential. The action potential doesn't vary from one action potential to the next. It is simply a signal that is like turning on a switch, and it's going to be the same every time, unlike what's going on over here. So for those of you who are old school and know about analog versus digital, this end of the neuron is the analog end of the neuron, and this end of the neuron is the digital end. So for those of you who are audiophiles or anything like that, then the digital end, or computer nerds or whatever, the digital end of the neuron is the axon, and the analog end would be the cell body and the dendrites. All right, now let's look at different types of neurons that we have. So what we've shown so far is what we call a multipolar neuron. And that means that we have a bunch of dendrites and we have one axon. Typically, most neurons have one and only one axon. Now, that axon may branch and send what we call collaterals to multiple targets. But nevertheless, there is one and only one axon. So whatever this cell decides to do, if it sends a signal, it will send it down its axon. And if there are collaterals that branch to multiple targets, let's say that this is a motor neuron, and its axon will innervate several muscle fibers, then that axon will have collaterals going off to each of those muscle fibers. But in any case, one action potential will innervate all of those muscle fibers. So each cell has one axon. Some cells have many, many dendrites. In fact, some cells have so many dendrites and so many connections with other neurons that we call this the dendritic field. And dendrite comes again from that term dendron, which means tree. So the dendrite can have connections with other neurons that may be signaling it, or a receptor which may be signaling it with information about senses. But in any case, some cells don't have a lot of dendrites. Some cells have only one dendrite. But in a lot of cases, most of the cells that we'll be talking about are what we call multipolar and have lots and lots of dendrites, but only one axon. Now, if we look at some other cell types, here's our multipolar cell. Here we have what we call a bipolar cell, which has one dendrite and one axon, and the cell body in between. Then we have something called unipolar or pseudo-unipolar, where basically we have a dendrite up here that's immediately fused to an axon, and the axon travels all the way to its target, and the cell body just sits off to the side. And we will see that almost all the sensory neurons in the peripheral nervous system are of this pseudo-unipolar type. So what we will have is we will have some kind of receptor up here. In some case, the dendrites are the receptors, and they will bring some information about sense. Could be pain, touch, temperature, what have you. And it will bring that information via this axon to a center that will process that information and either relay it on to somewhere else. So this part will come into the central nervous system right here, into the spinal cord, into what we call a nucleus. And then that information will be relayed to some other part of the central nervous system somewhere in the brain. And then that information will be used to make decisions. So almost all of our neurons within the peripheral nervous system, especially that will bring what we call our general senses, will be of this type, our pseudo-unipolar neuron here. Now, let's talk a little bit more about our neuron types in terms of what type of information they convey. We have sensory neurons. These are our afferent neurons. These are going to be bringing information from the periphery. And these could often be things like touch, pain, temperature. These are what we call general senses that are distributed all over our bodies. Or we could have some neurons that are very specialized that are bringing information from receptors in our eyes, for example, or our ears, maybe about sight, maybe about hearing, maybe about balance and equilibrium, maybe about taste, it's on our tongues, or smell from our nose. So we have two different types of senses. We have what are called our general senses, which are distributed all over our bodies and are things like pain, touch, temperature, vibration, these kinds of things, as well as proprioception, which is where our joints are in space. And then we have our special senses, and our special senses are located in special sense organs within the head. So all of our special senses are in the head, in organs in the head. So don't miss that on a test. So these would things like vision, hearing, balance and equilibrium, that is being able to keep your balance and not fall over. We also have our taste and our smell. These are all special senses, and they all reside within the head. Your general senses are distributed all over the body. And these sensory afferent neurons will be bringing information from these sensors to the central nervous system. Then we're going to have something called interneurons. And these are within the CNS, the central nervous system. And these will basically collate and pool together information and decide what to do with it. So we have interneurons arranged in pools, which basically are like processing centers. And these neurons will process information, decide what to do with it, and then maybe innervate a motor effector or a motor neuron. And a motor neuron is efferent. I should have said a motor efferent neuron, not a motor effector. The motor neuron is what will stimulate the effector. And this will conduct an impulse from the central nervous system via the peripheral nervous system to an effector that could be muscles, glands, adipose tissue, and the like. So here's an example. Here we have our division of our central nervous system and our peripheral nervous system. So we've got a sensory receptor of some sort that's going to convey some kind of information. And then that receptor will innervate a neuron. And that neuron will send the information from the receptor to the central nervous system. We could have several interneurons in a relay up to the brain or some other part of the spinal cord that's going to process that information. And then that interneuron will stimulate a motor neuron, which will leave or exit the central nervous system, and it is a motor efferent, and it will innervate an effector. So an easy way to think about this is a motor neuron is efferent, it exits the central nervous system, and it stimulates an effector. So that is basically an easy way to remember it. All right, next we're going to talk about the synapse. And this is how neurons will communicate with one another. And we'll talk about the synapse in the next segment.